Hello everybody, it's Mrs. Pound, and I'm back with another video for Chapter 4, Cellular Processes from BJU Biology 4th edition. We're in Section 4B on cellular metabolism and protein synthesis. And what I want to talk about today is protein synthesis from subsection 4.4. So our objectives today are going to be to describe the structure and function of the three kinds of RNA, differentiate between introns and exons, and list and describe the steps in translation. So the first thing we have to discuss is what is metabolism? Metabolism is the sum of all the reactions that happen in a living thing. There are all kinds of chemical reactions happening inside of living organisms. And just a few examples are photosynthesis, cellular respiration, and anything involving growth and movement. Everything basically involves metabolism and chemical reactions. So I want to first talk about the types of RNA because these are crucial for protein synthesis. The first type of RNA is messenger RNA, or shortened to mRNA. This is an RNA molecule that carries the DNA code for a polypeptide chain of amino acids from the nucleus to the ribosomes by passing through a pore in the nuclear envelope. So to explain this a little bit, the RNA and RNA is a single strand, as we can see in the background here. It looks almost like half of a DNA. DNA is double-stranded. RNA is only single-stranded. Um, a polypeptide chain is just a protein chain, and proteins are made of amino acids. And it's called messenger RNA because it's taking the message from the nucleus, the DNA in the nucleus, out to the ribosomes where proteins are made. And it does that by passing through the nuclear envelope, also called the nuclear membrane. And there are tiny pores that the RNA can go through to get out that the DNA cannot get through. Now, on the messenger RNA is called what is called a codon, and this is a triplet of bases or nucleotides that forms the code for a particular amino acid on messenger RNA. So, a codon, the cool thing about the code for the proteins is that every three letters codes for a different amino acid, which are the subunits of the protein. So transfer RNA is another type of RNA, abbreviated as tRNA, and this is an RNA molecule that delivers amino acids to the ribosome, and they have kind of this cloverleaf type shape to them, and they will read the codon that is on the messenger RNA and deliver, they actually carry the amino acids and deliver them to the ribosome. They have what's called the anticodon. This is the triplet of nucleotides on tRNA that will pair with the codon of the mRNA to line up amino acids during protein synthesis. So right here, the codon is meeting the anticodon. There's also ribosomal RNA, rRNA. This is the RNA molecule that combines with proteins to form a ribosome, and it contains neither codons nor anticodons. And there are two subunits to it. There's a large subunit and a small subunit, and the messenger RNA fits in between both of these here in the ribosome when it is read. So making RNA. RNA contains exons. These are RNA pieces put together to form mRNA, and introns. These are sections of RNA cut out and not used in the formation of mRNA. And so we can see here the exons are kept, the introns are discarded to form the final RNA. So this would be what's coded from the DNA. And scientists don't know why there are these introns. They're starting to discover why. They used to call it junk DNA, but we're also discovering that things that we think are junk necessarily aren't. And so that DNA that is, or the message that is gotten rid of, the introns, um, they are finding that maybe the purpose is for gene expression on whether or not the gene is shown, okay? So a way to remember this is Exons are expressed, introns go in the trash. So that might help you remember exons are kept, introns go in the trash. Now, translation. This is the manufacture of an amino acid chain. So this is the process that makes amino acids. And so I have a picture here that we'll be taking a look at. Now, the three types of RNA work together to form protein. All three types of RNA are formed in the nucleus by the DNA molecule. mRNA is in the form of a long chain of nucleotides and moves into the cytoplasm. And so here it is, it's moved into the cytoplasm and it's attaching to a ribosome. Okay, the mRNA has attached right here. Ribosomes move along the mRNA molecule and read the coded message. So it's nestled right there between that large subunit and the small subunit, and it is being read. As the ribosomes move along the mRNA, tRNA brings in the amino acids in a proper sequence. So you can see that here. There's a tRNA matching up with the codon. So the codon is on the mRNA. There's an anticodon. It's matching it up. It has an amino acid here on the end of it that it's attaching to this amino acid chain, this polypeptide protein chain that it is attaching. So the amino acids are bonded to form polypeptides, which move into the endoplasmic reticulum to be transported to the Golgi apparatus. Remember, this is why ribosomes a lot of time are on endoplasmic reticulum. That would be that rough endoplasmic reticulum because they are transporting proteins. And so here is that process again. So the mRNA attaching to the ribosome being read, transfer RNA coming in and matching up with those codons, with its anticodon, attaching an amino acid to the polypeptide chain. Now, just kind of an interesting fact here to throw in. Some genes for mitochondria are found in DNA inside the mitochondria. So we think that DNA is only in the nucleus. It's not. There is mitochondrial DNA. And the cool thing about mitochondrial DNA, here's another thing too, is they actually are using it to trace ancestry. So it's a modern tool that we're using to trace our ancestry, which is pretty cool. So, translation coding. How does this coding work? The nitrogen pairs are cytosine and guanine, just like DNA, C and G, and adenine and thymine slash uracil. A and thymine T, that's the matchup we know for DNA, but there's an extra nitrogen base pair, uracil, for RNA. So let's take a look at that. So the note, thiamine T is replaced by uracil and is a nitrogen containing base found only in RNA. So on RNA, instead of using T's, we write U's, okay? Which is pretty easy to remember because it's just the next letter in the alphabet. So I might give you a DNA fraction of DNA, a segment of DNA, and this is a DNA triplet. I will ask you to tell me what the codons are, the anticodons, and the amino acids. So let's take a look at this. So we know our matchup. So if it's a T, we're going to write an A. An A, DNA would be a T, but remember for RNA we use a U, so that's a U, and a C would be a G. So we end up with this codon, AUG, on the mRNA. So let's move down to the next one. So we have a G, what would we put? A C. A C would become a G, and an A would become a U. Remember, not a T, because it's RNA. A would be U, T, A, G, C. So UAC. Now try this one on your own and then I will show you the letters. So A becomes a U, C, G, T, A. Okay, now moving over to the tRNA. So we look at our mRNA And you should know the matchups by now, but A becomes U matches up with an A, and G with a C, so we have UAC. Now, go ahead and try to do the next one, and I will reveal the letters here. So we get GCA, then AUG, and ACU. Okay, so also notice this is a good check. If you look at the DNA and the transfer RNA, the code is the same except for instead of a T, there's a U. So that is a good check. Now, for the amino acids, to find the amino acids, we have to go over to the codon, the mRNA, and you will have a chart, and I will show you that chart right here. We're going to look at the mRNA, and we're going to find the code in the chart to find the amino acids. So here is the chart I will give you. It says amino acid and the genetic code. And the first thing I ask you to find, remember, we're looking at the mRNA, not the tRNA, is AUG. So if I look for AUG, I find it here. This is methionine. And so just go ahead and under the amino acids for that triplet, or for that codon, rather, write methionine. Now we're going to go to the next one, and it's CGU. Be careful you're looking at the mRNA, that you're looking at the codons. So CGU. And if I look in here, oh, there's CGU, arginine. So I'm going to write arginine for that amino acid in that blank. Then I'm looking for UAC. So UAC, have you found, oh, I found it. So tyrosine. Okay, so I write that. And then UGA. Oh, down here, stop. That's not an amino acid. That's curious, and we'll talk about that. So stop is what you will write. So let's go back to our chart. And here we have our chart. So we discovered by looking up our amino acids that the first one was methionine, arginine, tyrosine, and stop. So stop codons code for the end of an amino acid chain. Okay, stop codons, that is its purpose, to say, stop, we're done making the protein. And just as a reminder, proteins are used for growth, repair, and as an energy source. So there's some, looks like maybe steak on this plate. As the cow was growing, proteins were being made for growth. Cells become damaged all the time just from wear and tear, so they have to be replaced. And then they become an energy source for other things as food. So proteins can provide all of those things. So our objectives were to describe the structure and function of the three kinds of RNA, differentiate between introns and exons, and remember, introns go in the trash, and to list and describe the steps in translation. So remember to continue watching the video to answer the questions at the end, and don't forget your 5 questions in the margin.